In the previous video, we went through Morgan's crosses that allowed him to hypothesize about the phenomenon of sex linkage. Okay, so on the left here, we went through his first cross where we start off with a pure breeding white male mated with a pure breeding red female. And then after going through 2 generations of that cross, generating first an F1 and then an F2, Morgan set up the reciprocal cross here on the right, where he started off with a pure breeding red male mated with a pure breeding white-eyed female. And again, took that reciprocal cross to an F1 generation and then to an F2. Okay, the phenotypic results that you see here for both of these crosses allowed Morgan to hypothesize that white is a sex-linked gene. Specifically, it's on the X chromosomes. And let me take a minute to just kind of briefly summarize how sex is determined in Drosophila. So Drosophila have 2 types of sex chromosomes, an X and a Y, just like we have in people. The method of sex determination, how those chromosomal complements determine the sex of the individual, is different from how that happens in humans. But for now, let's just approximate that as just thinking that males will have an X and a Y chromosome, females will have 2 X chromosomes. Okay, that is typically the condition. So let's go back to these crosses and hypothesizing that white is found on the X chromosome. This white gene is found on the X chromosome. Let's see what we would expect in terms of the genotypes of these individuals, and if our expectations of genotypes are consistent with what Morgan observed in the phenotypes. Okay, so I'm going to zoom in here, and I'm going to focus on this first cross. Okay, there we go. So what we can do is first fill in the sex chromosomes that each of these, let's start with the parental generation, that the parental males and the parental females had. Okay, so we start off, the females would have 2 X chromosomes. The males would have an X and a Y. Okay. On each of these X chromosomes, the 2 in the females and the 1 X chromosome in the male, there would be a copy of this white gene. Okay, now the males, the parental males, have white eyes, so they would have a white allele. Okay, the red-eyed parental females would have a red allele on their X chromosome, and because red is the most common phenotype that we find in nature, we call that allele the wild-type allele. And in Drosophila, we use just a plus sign to denote the wild-type allele. Okay, so these red-eyed females, because the parentals were starting off with pure breeding parentals, so this red female is going to be homozygous for the plus or wild-type allele. Okay, so these are the genotypes we're starting off with according to this hypothesis. Now in the F1, the male will inherit an X chromosome from the parental female and will inherit a Y chromosome from the parental male. Okay, so the X chromosome here came from the parental female, so that would be an X plus, right? The Y chromosome here came from the parental male. And so because this F1 male has an X plus allele, he should have a red eye, which we see he does. Okay, the red females are going to get an X from each of their parents. Okay, from the parental female, this F1 female will get an X plus, and from the parental male, this F1 female would get an X with the white allele. Okay, so the F1 females are going to be heterozygous, right? The F1 males are not homozygous, they're not heterozygous, because both of those terms mean that an individual has 2 alleles, right? And this F1 male has only 1 allele for this X-linked gene, right? And so we need a new term here. The males here, we're going to use the term hemizygous, okay? Which means that they just carry 1 allele for this hemizygous gene. and they're going to express whatever allele they have, right? So the parental male is hemizygous for the white allele. He expresses that white allele and has white eyes. The F1 male is hemizygous for the wild type allele. He expresses that allele and has red eyes. Okay. This female in the F1 is heterozygous, has two different alleles. Okay. And because her phenotype is red, that tells us that the plus allele is dominant to the white allele. Okay. So complete dominance going on here. Now, I just want to make a note. The idea of wild type alleles, in this case, the wild type allele is dominant to the white allele. Wild type only means what is most common in nature. It doesn't imply anything about dominance or recessiveness. Okay, so it is possible to have recessive wild type alleles. It is possible to have dominant wild type alleles. You could have any kind of allelic interaction that you want. Wild type just means that it's the most common in nature. Okay, so we've established our genotypes in the F1. We've established that the wild type allele in this case is dominant to the mutant white allele. And let's see what happens when we cross this hemizygous red F1 male with the heterozygous red F1 female. Okay, so if we just scoot things up a little bit, I'm going to make myself a little bit more room. Okay, I think it might be easiest at this point to go back to a tool you're familiar with and use a Punnett square. Okay, so if we set up a Punnett square here, I'm looking at the cross of the F1s to generate the F2. Okay, so across the top, I'm going to put the alleles from the F1 mother, F1 female, I should say. So we have X plus, and we have X white. Okay, down the side, I'm going to put the alleles from the F1 male. So we have X plus, and we have Y. Okay, now let's fill in the field of this Punnett square and see what genotypes we expect in the F2. In this upper left quadrant, we have an X plus homozygote. That would be a female. And since we know that the plus allele is dominant to white, that would be, since it's homozygous, I should say, that would be a red-eyed female. In the next square, we have an X plus XW. That would be a heterozygous female. And just like we see in the F1 female, that would be a red-eyed female. Okay, down in the bottom left, we have X plus and Y. So that would be a male, and that would be a red-eyed male. Okay, and then finally we have XW and Y, and that would give us a white-eyed male. Okay, so you can see in our results here, we have 3 possible outcomes that give us red eyes. Okay, 1 outcome that gives us white eyes, and all of the females that are generated here, whether they are homozygous, wild type, or heterozygous, they're going to have red eyes. Okay, out of the 2 ways of making a male, half of the males would have red eyes, and half of the males would have white eyes. Okay, so that gives us our 1/4 of all the offspring are white, and all of those white-eyed flies are male. 3/4 of all the offspring are red, and they include a mix of both sexes. Okay, so that seems to make sense. This hypothesis seems to hold for this first cross, but if it's a good hypothesis, it's going to explain the reciprocal cross results as well. So let's look at that. Okay, I'm going to move over here and let's go through the genotypes that we would expect to see here too. Okay, so we have a parental type red male. He's hemizygous. He has an X and a Y chromosome, and his X chromosome is going to have a wild type allele. Okay, our pure breeding white female has 2 X chromosomes, and those chromosomes both carry the white allele. Okay, so when we cross this parental male with the parental female, in the F1 we get a white male, and because he's a male, he's going to have an X and a Y. Okay, he gets his X chromosome from the parental female, so his X chromosome is going to have a white allele, making him have white eyes as a phenotype. Okay, the F1 female is going to have two X chromosomes. She's going to get an X chromosome from the parental father, which has a wild type allele, and an X chromosome from the parental female that has a white allele. And as we see again, wild type in this case is dominant to white, and so this heterozygous female has red eyes. Okay, let's scoot this up a little bit. And I'm going to make myself some more room here. Okay, so again, let's use Punnett square to look at what we expect when we cross this F1 to generate the F2. Okay, so here's my Punnett square. I'm going to fill in the gametes that the F1s would produce. The females' gametes across the top, we would have an X plus and an XW from that heterozygous F1. The male's gametes down the side, we would have an XW and a Y from the F1 male. And then if we fill in these genotypes, first we're going to generate an X plus XW heterozygous female. Next we would have XW, XW, a homozygous white female. Down at the bottom we have X plus and Y, that would be a red-eyed hemizygous male. And finally we have XW and Y, that would be a white-eyed hemizygous male. Okay, so if we summarize these phenotypes again, let's look here. We have two outcomes that give us red eyes, 2 out of the 4, giving us half red. And you can see that of those red offspring, half of them are female. A heterozygous female would have red eyes, and half of them are a hemizygous red wild type male. Okay. On the other side of the Punnett square, we have two outcomes that give us white eyes, right? One of these two outcomes is a homozygous white-eyed female, and the other is a hemizygous white-eyed male. So half of the offspring would have white eyes, and they would include both males and females. Half of the offspring would have red eyes, include both males and females. Okay, so we can see from these crosses how Morgan came up with this idea of sex linkage, and that the inheritance of traits that often have nothing to do with the sex of the organism, right? Flies have eyes with colors, whether they're males or females, but they are the pattern of inheritance and the expected ratios in the F1 and the F2 depend on what the sex of the individual fly is.